Okay, part 3, here we go. We're going to start by talking about the cytoskeleton. Now, a lot of this that we're going to dive into should sound partially familiar because of the special structures PowerPoint that we saw before this. That special structures one actually should have come after this, but that's okay. It's all... I did a pretty thorough job of explaining it previously. We're just going to get a little bit more information on this part. So the cytoskeleton is responsible for shape and movement of the different organelles. We're going to look at microfilaments, intermediate filaments, and microtubules. Microfilaments, intermediate filaments. These are the three protein type structures that we're looking at when we talk about the cytoskeleton. Within the cytoskeleton, we want to know those functions. It has binding sites, it helps keep its shape, it helps with movement, and also helps to specialize the specific cells. We've seen here shows kind of the general overall type stuff of all this. Again, we don't need to memorize every single bit and piece of this. Let's continue going with kind of what we describe each component of. So microtubules, we touched on these already before. And these are the largest cytoskeletal element. They are hollow tube-like structures made up of protein subunits called tubulin. Tubulin, this is going to be on the test. Tubulin is a globular protein. So make sure we know that. Tubulin also has two different types of tubulin. There's an alpha tubulin and a beta tubulin, which forms a structure called a heterodimer. So when we take a single alpha tubulin and a single beta tubulin and we put them together, that is a heterodimer. So two different parts. These heterodimers are then put together. So here we have a bunch of heterodimers. They get put all together to create what's called a protofilament. And then we put a bunch of the protofilaments together and we eventually get a microtubule. This is very similar kind of when we're talking about like DNA to the nucleosome to then continuing further on to the chromatid and then to a chromosome. How it goes from the most basic, and as it combines and builds, it gets bigger and bigger and it's called something different. Same concept here. So those heterodimers turn into protofilaments and then become a microtubule. These may form as singlets, doublets, or even triplets. We actually saw this in that previous, when we were talking about the cell junctions and the special structures, how when we looked at some of the cilia who have movement and stuff, we saw that they had the 9+2 structure, where you have 9 doublets and then the 2 singlets in the middle. So this is just showing you can have single microtubules, you can have them in doubles or even triples. So the example for the triples is a centriole. Centriole has 9 triplets, and most cells have one pair of centrioles. And these will play an important role in cell division and cell mitosis when we get to that component later on. Here are microtubules, the tubulin subunits, protofilament being the structure of the microtubule, centriole with the triplets, and we'll get more into kind of that nomenclature, that kind of the description of the centriole when we talk about cell division in the next lecture. A pair of centrioles, which is what we have within a cell, is called a centrosome. So just some more vocab for you, make sure you know those words. There's also something called molecular rails. So the centrioles and the centrosome, these are going to play a key role in sending out kind of these fibers. It's a network of microtubules, so they send out and they extend these microtubules from the centrosomes, and it's going to go and it's going to link onto components of the cell itself, as well as some of the different organelles, as well as the chromosomes when they're forming, when the cell starts to split. And it helps to kind of pull and move everything into the proper position as the cell splits. And again, we'll see more of that kind of when that happens in the next lecture. this process of which is happening, where it's grabbing all this stuff and moving everything. They call it molecular rails or cross bridges because it follows like a rail system, like a train system. Here we go. Organelles get grabbed on by a motor molecule, and it literally pulls it along to put it into place, and it uses ATP to do that. There should be a video. Let me see if I can find that video really quick and add it on here. I don't see it here specifically. I'll see if I can find that video. If I can find it, I'll post it in here again. Now, mitotic spindles as well, kind of going along with this. The mitotic spindle is a structure composed of microtubules that plays a critical role in mitosis. Here, molecular rails are very similar to the mitotic spindles, just what their jobs are. It's a little bit different. The molecular rails, we're focusing on organelles and vesicles and those components. The mitotic spindle is essentially very, very similar, just it's going to focus on the division of these chromosomes. Both of them are made up of microtubules, just they give them different names because they have different functions. A lot of this, the rest of this kind of stuff is review, talking about the axoneme, the cilia, and the flagella, kind of all of that they do, what they're made up of, those 9 doublets and 2 singlets, all of those functions, internal, external functions that are all through here. Again, pretty much those same similar structures. And a lot of this we already saw before. Microfilaments, again, these are composed of actin. We already talked about the G-actin and the F-actin, kind of how that's all produced, how you have the single components, as well as going to the F-actin components. The G components are the individual parts, and they form together to form F-actin, then all links together to create a microfilament. Again, more of the functions. Mostly this right here, the functions component, and the composition of the function, this is really what gets tested. A lot of test questions tend to come from slides like this. Make sure, especially the functions, professors really want you to know what stuff does. So all of this stuff down in here, especially the unique ones, like the amoeboid movement or of phagocytic cells, that's going to be one that's going to be really important. That's going to come back. Again, microfilaments, made up of a protein called actin. It's a globular protein that forms a chain. The monomer is G-actin. The chain is F-actin. All together it becomes a microfilament. Here, again, these are those functions. Again, and actually, I think G-actin. Let me actually delete this slide because this has the exact same information. Sometimes I would do that if I was rewording the slide and adding more to it and making it a little bit neater. Sometimes I accidentally forgot to delete the slide before. Okay. Microfilaments, more about microfilaments. We have the microfilaments are polar, so you have a positive side and a negative side, and from the positive side you get polymerization or building of it. From the negative side you get the breaking down or the depolymerization of it. This is more just actions of that, some of those functions that we see here. More of like the muscle type cells in this motor muscle contraction. We'll look a lot deeper into that in Physiology 1 next trimester. Here the actin is part of the cell cortex, playing its role in the cellular structure. The cleavage furrow. So this is during what's called cytokinesis. We're going to talk more about this in lecture 3 material. Cytokinesis is the actual splitting of the cell. So when we talk about, this whole process with the molecular rails moving everything, and we have those mitotic spindles pulling stuff, we also get the microfilaments and actin playing a role in the actual splitting and kind of squeezing of the cell in order to break it into two individual daughter cells. That's called the cleavage furrow. We'll see that again when we talk more about, the actual mitosis process. Now, intermediate filaments, these are fibrous, not globular proteins. They're more stable, made of a bunch of different protein subunits in different cells. So they're made up of a bunch of different stuff depending on what cell you're actually looking at. Their main job is, again, to provide more of the structural strength in a bunch of different types of cells. So here we can see, again, you don't need to really memorize this whole thing. The one that typically does get asked about is keratin is in the skin. Later on, we'll talk about more about that as well. But you can read through it, just kind of look at some of the names. Don't worry too much about this specific slide. Here's, again, just more kind of pictures. Medical application. Did we talk about? Must be in the next couple PowerPoint. We did. Yeah, it's in. Sorry, it's next. Is lecture 3 material that we jump into college in. So we'll jump into that later. Just the intermediate filaments reminded me of that. So, but that's not till next lecture. Okay, so the example of medical application. So some inhibitory compounds used in cancer chemotherapy block activity of the mitotic spindle in rapidly growing tumor cells. So essentially, we'll see this more when we look at mitosis. Essentially, mitosis has a bunch of checkpoints within it to check and make sure that the cell should be dividing, that it's healthy enough to divide. When we have cancerous cells, essentially we skip those checkpoints, or we just blast right through the checkpoints without the cell actually being checked, which is why it grows uncontrolled, which is what we get, cancerous tumors. So part of chemotherapy can actually block specifically the mitotic spindles to stop the growth of the cancer, allowing the body, to recognize, hey, these are not good, and our own immune system to destroy some of those cells. Finally, this one, class 5 intermediate filaments defects are found in muscular dystrophy, which is a group of genetic diseases causing progressive muscle weakness and degeneration. We'll talk a lot more about muscular dystrophy in future pathology courses. This is kind of the flow. This really should be towards the end of the kind of endoplasmic membrane system. It should be paired kind of with all of that, but there wasn't a great place to put it with all of that. So here it is, this last little portion. This just kind of goes over the flow of how everything flows. So the nucleus, we get mRNA transcription. That mRNA goes into the rough endoplasmic reticulum, which it translates it. We'll learn a lot of these words later. Transcription is copying the DNA. Translation is reading the mRNA as with the ribosomes in order to produce the proteins to make the polypeptide chain. And then we go to transport vehicles in the Golgi. The Golgi modifies it, packages it, and ships it. And then we get lysosomes inside of the cell itself, or it goes to the outer membrane. And again, a little bit more information is here down below. So this whole pathway, kind of where everything is going with the endomembrane system. Okay, and that is it for cell structure. A good amount of material here. Some of the next couple of slides aren't as long, so we'll try and do them in less parts. But thank you so much for watching. As always, if you're interested in getting any of the notes and everything that I have available, or as well, if you want more information on specific subjects or specific areas, feel free to reach out, leave a comment asking for specific information, and I will see what I can put together in order to offer and include with all of the stuff that I do offer. Again, leave a like on the video if you can. It really helps out, and have a wonderful day, and good luck studying.